Hi everyone. Today's topic is the phage Lambda, or bacteriophage Lambda, known to infect E. coli cells. This wonderful genetic system has been a source of inspiration for many biologists and non-biologists. It has motivated many scholars to move from their domains of study into biology. This wonderful gene regulatory network controls the outcome of the E. coli action by the phage Lambda. So the phage can grow in a lytic way, ending up with the lysis of the bacterium and the release of the phage particle. However, the phage has another survival strategy during which the viral genome gets integrated into the E. coli chromosome, and then it's going to be transmitted in a stable way from generation to generation. This bistable genetic switch is amazing in that sense. The system can rest on either on or off on either pathway. And this switch is controlled by key regulatory proteins. We will shed light on all of them, but it's highly dominated by stochasticity, which means that two identical cells growing under similar conditions and infected by the same phage can have different outcomes, which means that stochasticity can drive the phage into one or the other pathway. I remember I was having lunch with three of my colleagues from the Lambda laboratory in Gothenburg, Sweden, when one of them told us a joke about the phage Lambda. It was about a question he raised during the exam. The question was, what direction would the infection of E. coli cells by the phage Lambda take had we had a mutation in the gene coding for C2, which is one of the key regulatory factors? And the student answers to the right direction. And actually, the joke was meant to be funny, and it was funny. However, at the very moment, I couldn't help think that the student was somehow right. He did not elaborate on the answer when he said to the right direction, but he was not totally wrong. Let me show you. So it's good to remind you that the phage Lambda has been a model for many years, leading to key discoveries in the field of molecular genetics, mainly unraveling many aspects of the process of regulation, recombination, as well as transcription, initiation, and termination. I'm starting here the presentation. Let me just say first that I am Professor Fahad Nasser from the Lebanese University in Beirut, and down you have my website, private website. So let's start our presentation today, which is about the phage Lambda, sort of a genetic miracle. So the phage Lambda, as you can see on the screen, is made up of the head and the tail, in addition to the absorption plate through which it binds to the cell wall to inject the DNA that is confined to the head. So this is the structure of the phage Lambda, as you can see. And here I would just say quickly that following the infection, as you can see, the phage Lambda bound to the E. coli cell membrane injects its DNA molecule, and after that we have two possible survival pathways, as I told you previously. So the phage can grow lytically, as it's shown on the right, where the genome is going to be replicated at the head and tails. Proteins will be produced and assembled, and then mature phages will take shape, and the process will end up with the lysis of the bacterium and release of those virions. The other survival depending on the conditions and many other factors we will speak of, the injected viral genome will integrate the E. coli chromosome, forming a stable relationship. This linear form we refer to as prophage, and it's going to be transmitted in a stable way generation after generation. And you can see that following this infection, and if the lysogenic pathway is favored, we have production of this repressor protein that maintains the prophage confined to the E. coli chromosome and confers immunity to the cell that we call lysogenic cells against infection by the same bacteriophage later. However, while lysogenic bacterium can divide for many generations, maintaining the prophage in this locked state, there is possibility under some conditions for the phage or the prophage to get released and then to follow what we call the lytic pathway, a process that we refer to as induction. So you can see very well here we have a bistable switch, a switch between two pathways, the lytic to the right, the lysogenic to the left, and this bistable switch is stable. So the cell fate following the infection will depend on a number of factors, mainly, in fact, the environmental conditions as well as the genotypes of the E. coli cells and the phage. The first step after the infection and the injection of the viral genome, this DNA molecule will circularize to the sticky ends. These 5' overhangs we refer to as the COS sequences. So the interaction or the base pairing between those sequences will generate a circular DNA molecule. Naturally, the gap will be sealed by DNA ligase. So this is the initial step after the infection, the circularization of the phage chromosome. Now, if you look at the map of the phage, it's very complex. So here, the linear map showing the different key regulatory genes. I will show you this later. However, it's divided into region that we call regulation and then recombination versus DNA replication. And we have those genes that are dedicated to the head and tail, so head and tail morphogenesis. And we have two more that are concerned with the lysis of the bacterium at the end of the lytic pathway. I will show you the map in a linear way to start, in fact, the gene regulatory network and show you what are the decisions that should be taken by the phage after the initiation of the infection. And here you can see something beautiful, the linear map that is bounded or flanked by the overhangs corresponding to the COS sequences, which are involved in the circularization of the Lambda chromosome after infection. So you can see the region we refer to as regulation, and we will focus on this region on the coming slides to show you, in fact, how the decision at the very beginning of the infection is going to be taken by the phage Lambda, something very beautiful. So we have the regulation, and then to the left you have recombination, and we have the head and tail genes for the head and tail morphogenesis. To the right of regulation, we have the gene O and P, both concerned with the replication of the viral chromosome. And we have the two genes I told you about, the S and R, both are concerned with the lysis of the bacterium. So if we have a close-up look of the regulation region, you can see very well that we have here key regulatory factors encoded by the gene C1, CRO, N, and to the left of N we have C3, to the right of CRO we have the C2 genes. So these are the main key regulatory factors that will control the cell fate of the phage Lambda infecting E. coli cells. So, in fact, if you look at the regulation region, you will see two promoter that we call PL and PR. PL is a promoter to the left that control the expression of N and all the genes to the left of N, and we have PR, the promoter to the right that controls the expression of CRO and all the genes to the right of the CROs. So in addition to the promoter, you have operators, which are the binding site of the repressor. I will move slowly, I will show you. Now, if the phage lambda chooses, in fact, the lysogenic pathway, as I told you, the first step is going to be the integration of the phage chromosome into the E. coli chromosome through site-specific recombination. So on both, in fact, molecules, the viral genome as well as the E. coli chromosome, we have two sites we call attachment site. So we refer to that site on the phage lambda chromosome at P and at B for the equivalent site in the E. coli chromosome. So interaction between these two sites, site-specific recombination involving these two sites, will see the integration of the phage chromosome into the E. coli chromosome, leading to the formation of the prophage. Oh, very well, if we come back to the map and starting with the lytic pathway in details. So again, after infection, we have a bistable switch where the system will rest on either one of the two possible outcomes. So one of the two pathways will be activated, the lytic or the lysogenic pathway. Let's start with the lytic or the lytic pathway in details. So again, we focus on the region. The first step is the expression of the gene N by the cell machinery, by the normal RNA polymerase of the E. coli cells. So it binds to the PL as well as PR. So from the PL and PR, we have expression of the first two regulatory proteins, N to the left and CRO to the right. This, in fact, step will end up with the first terminator to the left that we call TL, or the first terminator to the right that we call TR1. So this expression will generate the first two proteins N and CRO. N is an anti-terminator, which means when N is available, it will bind to specific sites we refer to as NOT. So we have two. One we call NOT L, which is a site to the left, and the other is NOT R, a site to the left, to the right. So both of them are located downstream of the promoter, PL and PR respectively, when N becomes available. And when N binds to the corresponding NOT sites, it will modify the RNA polymerase in such a way to ignore the termination sites and keep going downstream to the left of N and upstream to the right of CRO, generating what we call two polycystonic messenger RNA. And as you can see, starting with the left transcripts, so we have here N in addition to C3, which is the other regulatory protein, in addition to all the genes of the recombination region, a long polycystonic RNA. For the right transcript, when we have transcription rightward, we have production of another polycystonic RNA molecule that entails, in addition to CRO, the C2 regulatory protein, in addition to O and P genes concerned with the replication, and one more regulatory factor that we call Q. As you can see, Q on the bottom. This is another anti-terminator. When Q becomes available, a Q will bind to a specific site that we call cut, in fact, downstream of Q. And then when we have initiation of transcription from the second promoter to the right, we refer to as PR prime. In the absence of Q, usually, this initiation of transcription will generate a small RNA molecule without any coding capacity. However, when Q, the second anti-terminator, becomes available, what will happen is simple. So this Q will change somehow the conformation of the RNA polymerase, and such modified RNA polymerase will ignore the terminator to the right, the second terminator to the right, TR2, and keep going downstream, leading to the expression of a long polycystonic RNA, including the lysis genes, in addition to all the genes concerned with head and tail morphogenesis or the virus morphogenesis. They are put downstream, in fact, of the PR prime due to the circularization of the viral genome. And that way, we have expression of the lysis as well as the head and tail genes. So we have production of the head and tails proteins, their assembly, and then naturally we have replication of the genome and assembly of the mature particles. Something very beautiful. So we have 3 steps, starting with the expression of N and CRO. We call those genes early genes, and then we have the middle genes corresponding to the recombination as well as the recombination and replication genes together with the second anti-terminator Q. And we have at the end, in the 3rd step, the expression of the later genes thanks to the Q anti-terminator in a long polycystonic RNA from PR prime, as you can see. Very well. So when the O and P gene products are available, we have replication of the viral genome, starting with the Kern's mechanism, where we have duplication of the genome after each round. However, we have switching from the Kern's mechanism to the rolling circle mechanism, which is a very efficient mechanism, as you know, that generates a concatenere of genomes linked head to tails. So after that, naturally, those concateneres will be processed by the complex encoded by the viral genes NU and A. So this complex will cut at the Cas sequence, generating one single genome. This genome will enter the head. So the head is said to be mature. Then the tail will join to form a mature particle. That way, all the mature particles will be ready to be propagated or to be disseminated following the lysis of the bacterium. Again, if you come back to the lysogeny in details, so you will see very well that the initial steps are shared by the lytic and lysogeny pathways. As a matter of fact, you can see very well the regulation region, the expression of N and CRO as the first step, which are the early genes by the RNA polymerase, the normal RNA polymerase, not modified. And then when N becomes available, the first anti-terminator will change the RNA polymerase to drive the expression of 2 long polycystonic RNA to the left and to the right. So again, we have here those polycystonic RNA that includes to the left of N C3 and to the right of CRO C2. So we will have to focus on these 2 regulatory proteins. So once C2 becomes available, so C2 will induce the expression of the phage lambda repressor C1 through the promoter for repressor establishment, as you can see here, where I'm showing you. So the C2 will bind to the site within the promoter and induce expression of C1 from a different strand. As you can see here, this transcript in red. So we have expression of C1, and when C1 becomes available, C1 will induce its own expression from another promoter that we call promoter for repressor maintenance. Naturally, the expression from the promoter of repressor establishment is more efficient, leading to the production actually of more C1 proteins. But once C1 binds to the region here, upstream of the promoter for repressor maintenance, we have sustained expression of C1 to, in fact, autoregulate its own production. And when C1 is available, so C1 will bind to the operator, to the left and right operators, and block the expression of CRO and N. And as a consequence, no Q will be produced, and the later genes will not be expressed, which means that lysogeny is favored. So the genome that is circular will integrate the E. coli chromosome. Something very beautiful, as you can see. So again, here, the key factor for the production of C1 is C2. And the role of C3, if you raise a question, is going to be to stabilize C2, preventing its degradation by cell proteases. So C2 will induce expression of C1 from promoter of repressor establishment, and later the expression of C1 will be autoregulated by its own product, so to maintain the repressor in the right concentration to block further expression of the other genes that are essential for the lytic pathway. That way the system is locked, and then the circular genome will integrate the E. coli chromosome. So we have many questions that are still in the dark. We will answer them, starting with the promoter to the right and to the left. So both of them are overlapping operator regions. So we have an operator to the right and another operator to the left. And each is split into 3 regions that we refer to as origin 1, origin 2, origin 3. And if you look very well, you can see very well that the promoter to the right is overlapping the operator region 1 and 2, while the promoter for repressor maintenance is overlapping the operator region 2 and 3. We have the similar, in fact, structure for the operator to the left, with operator, in fact, regions 3, 2, and 1. And as you can see, the promoter to the left, which is concerned with expression of the anti-terminator N and the genes of the recombination region, is overlapping at least with the operator region 1 and to some extent with 2. So now you can see very well that when C1 is available, it will bind to the operator, preventing the expression of an N-CRO with differential sensitivity or affinity. In fact, C1 binds with higher affinity to the operator region 1, to the right, and 1 to the left. That way, it will prevent the expression of an N-CRO. While the binding of the C1 to the operator region 1 and 2 will enhance its own expression. Let me show you. So here you have the transcription in the control region in lytic and lysogenic pathways. So for the lytic pathway, we have expression of N and CRO from their promoter, the PL and PR respectively, while C1 should be blocked. While during lysogeny, somehow the C1 should become available and block the expression of the N and CRO from their PL and PR promoter respectively, and enhances its own expression to maintain the system locked. Very well. So if you look at the C1 repressor structure, it has a modular structure with 2 domains connected by a linker, a flexible linker. So we have the N-terminus domain featuring the DNA binding part and the activation region. So the C-terminal domain features dimerization as well as tetramerization structures. So the C1 repressor acts as a dimer, and this dimer shows affinity first to the operator region 1, higher affinity to 1, less to 2, and less to 3. So we have differential affinity, which means that when C1 is expressed first, it will block the expression of CRO. But when we have plenty of C1, initially it will bind to the other operator region and end up with blocking its own expression. So it's sort of autoregulation, a classical autoregulation. So in fact, the promoter for the repressor maintenance is weak. And it's well known that the binding of the C1 repressor as a dimer to the operator region 1 and 2 will enhance its own expression thanks to the activation region that is shown previously. So the C1 repressor is enhancing its own expression by assisting the RNA polymerase in the initiation step. When it comes to the CRO, it has a normal structure with single domain, but still act as a dimer. This dimer has higher affinity to the operator region 3, which means that under conditions that favor the production of CRO will bind to the operator region 3, hiding the promoter for repressor maintenance and preventing the expression of C1. That way, the lytic pathway is favored. And when we have plenty of CRO dimers, you can see here that the binding will extend to the operator region 2 and 1. So again, the expression of CRO is subject to autoregulation, which means that CRO will block its own expression when we have the right concentration. So I would like to add at this level that CRU, in fact, promoter is already strong, which means that the RNA polymerase has good affinity to the promoter and doesn't require an enhancement, which means that the binding of a CRU as a dimer to the operator region 3 is not enhancing its own expression. So it doesn't require any assistant at this level. And here I'm showing you both, in fact, patterns of expression during lytic and lysogenic growth. So on the top, you have the lysogenic cells showing the C1 repressor as a dimer on the operator region 1 and 2, enhancing its own expression so to maintain the good level of C1 repressor. And on the bottom, you have the lytic growth, where the CRU dimer bound to the operator region 3 will block the expression of C1, which is essential for the pathway to keep going rightward. And here, the student was right when he wrote to the right direction, because when C2 is inactivated, naturally C1 is not produced. The CRU will be able to gain access to the operator region 3, preventing C1 from being expressed. And as a consequence, we have here expression to the right for the production of the Q, the second autoterminator, which is essential for the production of data genes. And naturally, this will favor the lytic pathway. So the student was in part right when he wrote this funny answer to the right direction. Now the C1 repressor, in fact, binds to the operators in a cooperative fashion. So after the binding of 2 dimers to the operator region 1 and 2, similar to the operator region, in fact, 1 and 2 to the left. So we have tetramerization, and this tetramerization will lock the system entirely, preventing further expression of C1. So this is the autoregulation we spoke about before. But in addition to that, the tetramerization will lock the system, preventing any expression from the PL and PR promoter. So the system is completely locked. We have to revisit the integration of the viral genome into the E. coli chromosome as an essential step during lysogeny. So if C1, in fact, becomes available and blocks the expression of the second autoterminator, naturally, we favor the lysogenic pathway. And here, for the integration, we need the production of another gene, the gene int, coding for integrase. So in this, in fact, step, we are focusing on that issue, the integration of the phage genome into the E. coli chromosome, leading to the formation of this linear form, or we call prophage. From that point on, the cell will gain an immunity against infection by cellular phage, thanks to the repressor C1. However, the issue is how the int gene is expressed. The gene int codes for the integrase, which is required for the integration of the viral genome into the E. coli chromosome. However, during the lytic pathway, that expression should be prevented. As a matter of fact, we are focusing here on 2 genes, the gene int and xis, coding for the integrase and exogenase, respectively. The gene int is required for the integration. The gene int and xis are both required for the release of the phage lambda from its chromosomal lock during the induction process, when the cells, in fact, are somehow under conditions that favor the release of the phage lambda to follow the lytic pathway. So how the int gene is expressed? First, during the lytic pathway, no int gene should be produced. And as a matter of fact, it's not, because the int gene is part of the long polycystonic RNA that is initiated at PL. And this PL-initiated transcript will end up in a region that we call B region, that feature a sequence that we call SIB, that can adopt what we call a Herpin structure. This is an unstable transcript that is degraded. As I will show you on the next slide, this Herpin is a substrate for RNA polymerase, for the RNA ribonuclease. So what happens during lysogeny? During lysogeny, in addition to this long polycystonic RNA, including int, the int gene is expressed in a different way thanks to the C2 in fact, the gene product. C2, in fact, enhances the expression of the C1 repressor from the promoter for repressor establishment, but it has another effect. It induces the expression of the gene int from a promoter that we call PI. It's a promoter specific for the gene int. And as a consequence of this initiation that is normal, driven by a normal RNA polymerase without this modification, so the transcript or the transcription process will end up at a genuine terminator, leading to a normal transcript that is stable, and that transcript will be translated, leading to the production of N of the integrase, and that integrase will drive the integration of the phage lambda into the E. coli chromosome. Something very beautiful. Let me show you again. Here you have the map. We are focusing on the PL promoter for the expression of N, C3, and all the genes to the left of N, mainly the recombination. And we are here focusing on the cis and int. And you can see very well, downstream of int, we have the attachment site. Remember this fact. We have the attachment site, the attachment P, that is involved in site-specific recombination during the integration step. And downstream of the attachment P, we have a region that we call B, within which we have a sequence that we call SIB. And that SIB will adopt this herpin structure I have spoken about. So the initial step is the production of N, and then we have a long polycystonic RNA initiated from PL in presence of N, and that transcript, where we have modification of the RNA polymerase, will ignore most of the termination sites and keep going downstream of at P into the region B, and maybe even further. This transcript is unstable and will be degraded. So as a consequence, there is no expression of int or cis, and this is exactly what is required for the lytic pathway. None of them is needed if the lytic pathway is favored. So transcript 1 that is initiated at PL is an unstable transcript that will be degraded by ribonuclease. While if somehow the lysogenic pathway is favored, thanks to the production of C2, and C2 is stabilized by C3 under some conditions, like for instance, high concentration of cyclic AMP, for instance, when glucose is exhausted, let's say, and this will depend as well on other factors that could be the size of the cell. So here we have a transcript that is initiated at PI, and this initiation is enhanced by C2. So this normal transcription initiation at the terminator that you can see very well here, upstream of the at P, this normal transcript will be translated as stable, leading to the expression of the intergene and the production of the integrase, which is required for the integration of the phage lambda into the E. coli chromosome. And you can see here side by side the SIB sequence. In the transcript number 1, as I told you, here we have a different structure that is a substrate for the ribonuclease 3, so it's going to be degraded, and degradation from 3' end will keep moving up, in fact, in 3', 5' orientation, so no integrase or exogenase are going to be produced. While for the second transcript, transcript number 2, it features a normal intrinsic terminator. You can see very well the herpin on this tail of U. So this is a normal terminator, and RNA transcript is stable, so we have production of the integrase. It's normal. The integrase is going to be produced only if lysogeny is favored. So we have transcript number 2, and under conditions that favor the lytic pathway, naturally, we have production of this long polycystonic RNA that ends up in the B region, where we have here the formation of this herpin that is a substrate for RNA3, and then transcript 1 will be degraded. And now we are getting to the end, how to make the right decision between lysogeny and the lytic pathway. I told you that we have some key regulatory factors, mainly the C2. You remember C2. in presence of C2, if C2 is stable, not degraded by the host proteases, then C2 will induce expression of C1 and the gene int. And when C1 becomes available, and depending on which one gains access first to the operator, CRO or C1, we have here a switch or a choice between two different options. So conditions that favor the lytic pathway, let's say CRO becomes available in the right threshold, it binds to the operator OL and OR and blocks expression of C1, but transcription will keep going on to the right. So P, in fact, R will be used. Some transcript will keep going for the production of Q, the second terminator, which is required for the expression of later genes, the genes of lysis and morphogenesis. Under conditions that favor lysogeny, let's say poor conditions where the concentration of cyclic AMP is high, so together with C3, C2 is stabilized, and C2 will induce expression of C1. To some extent, we have high amount of C1. C1 will bind to both operators, enhancing its own expression, but blocking the expression of N and CRO. So that way, we favor the lytic pathway. So under, in fact, these conditions, I would say the decision to either pathway, the bistable switch will depend on which one, the CRO or C1, will gain access first to the operator. And that step will depend on whether C2 is stabilized and becomes available, in fact, to induce expression of C1. So we still have one more issue to go on. In the old model, they state that C2 is essential, in fact, for the production of C1, and the switch or the bistable switch will depend largely on C2 production. However, the new model have shown that it's a question of threshold, and the absence of C2, for some reasons, the Q appears sooner and in high concentration, which means here we have a sort of competition between C2 and Q. And you can see very well which ones of the two factors, C2 or Q, will be able to go beyond the threshold. One of the two pathways will be favored. So if we get to the threshold with C2, we have lysogeny. And if conditions favor the production of Q and Q goes beyond the threshold, then we have the lytic pathway that is favored. So as you can see very well that the bistable switch is controlled by key regulatory proteins, and here you can see very well the role of C2 and a Q. However, it's dominated by stochasticity. The slightest variation in the metabolism could favor, in fact, one or the other pathway, depending on which one of the two factors, C2 or Q, will be able to go beyond the threshold. We still have one more issue. When the phage lambda is locked into the E. coli chromosome, there is possibility under some conditions to get excised, to get released from this lock and then to follow the lysogenic, the lytic pathway, a process that we call induction. And what are the treatments that might induce, in fact, the phage lambda? One of the regulatory mechanism, global regulatory mechanism, we refer as the SOS response. In fact, this response is concerned with the survival factors that help the cells withstand some treatments that could damage DNA or block DNA replication. All these treatments will converge into the gene REC-A, and once REC-A is activated, then for the SS response, we have a key regulatory protein that we call LEX-A that blocks the expression of all those genes coding for the survival factors, will be induced to cleave itself. LEX-A inactive, all the genes here of the SS system will be expressed, and those genes, as I told you, will help the cell survive, repair the damage, and then overcome the consequences of the treatments. And once, in fact, the damage is repaired, naturally, the signal will drop, the REC-A will be inactivated, LEX-A will be able to regain access to all the promoters of those SOS genes, and the system will come back to normal. However, one consequence of the activation of the SOS response is the inactivation of C1 in a similar way to LEX-A inactivation by activated REC-A. Let me show you. Following the activation of the SOS response, we have here induction of the REC-A or activation of the REC-A, and REC-A will cause the C1 to cleave itself in a similar way to LEX-A. And once C1 is cleaved at this connector region, so it can no longer dimerize, and the inability to dimerize will inactivate the C1 repressor. Following the inactivation, the lytic genes will be de-repressed, and naturally the lytic pathway will be activated, ending up with the lysis of the bacterium. And you can see here the structure of the C1 and the region between the N-terminus and C-terminus region corresponding to DNA binding and dimerization, as well as tetramerization domain. The connector will be cleaved. And as I told you, once it's cleaved, the C1 repressor is unable to dimerize and loses its function. That way, it will release the operator region 1 and 2, which favor the expression of CRO. And as a consequence, we have expression of the latent genes, leading to the production of the head and tail proteins for their assembly, as well as the lysis genes. We'll say here we have one more issue to be concerned with. For the release or the induction of the phage lambda, we need the product of int and exis together. But again, here we have production of these two genes from the PL-initiated transcription. But if you remember, now we are dealing with the prophage. The prophage that has integrated the E. coli chromosome thanks to the site-specific recombination involving ATP, which means that all the genes of the head and tail morphogenesis will be brought, in fact, to the other side. And the PL-initiated transcription will end up in a normal region without the SIB sequence, and this transcript will be stable, leading to the expression of both the integrase and exogenase, which are required for the release of the phage lambda from its chromosomal lock, what we call prophage induction. And then we have circularization, and naturally the lytic pathway will be followed till the lysis of the bacterium. So you can see everything is fine-tuned. The question comes here about the co-evolution and adaptation of the phage lambda to the E. coli cells. So once I mentioned the stochasticity, so it might be here, in fact, exaggerated to some extent, because the evolution of the phage lambda or co-evolution of both partners led to the fine adaptation to the slight change in the metabolism or the growth conditions. For instance, if you come back to the questions of LEX-A and the C1 repressors being inactivated by cleavage thanks to the activation of REC-A following those treatments that damage DNA. In fact, the response of these two factors to the damage is completely different. LEX-A has evolved to answer the slightest, in fact, signal, the slightest damage. So naturally, it will be cleaved quickly, and the system will be induced, and the damage will be repaired. While the phage lambda C1 repressor has evolved to answer only those level of damage that endanger or threatens the cell survival. Under those conditions, only the cleavage of C1 repressor will proceed to completion, leading to prophage induction. Under small damage, the phage lambda C1 repressor will not be destroyed completely, and the phage or the cell will restore normal growth after a while once the damage is repaired. So this is, in fact, this kind of differential sensitivity of LEX-A and C1 to damage explains how the adaptation of the phage lambda to the E. coli cells has evolved over the years in fact, to adapt to the slightest, in fact, change in the cell metabolism or growth conditions. So with this, we have come to the end of this, in fact, the beautiful story about the phage Lambda. I'm quite sure you're going to have many questions, but if you go through the presentation again, you will see the beauty of the system and how every single, in fact, decision is dominated by some factors under specific conditions. So I did not elaborate further on the stochasticity as well, on the different, in fact, different aspects of the gene regulatory networks. I believe it's quite complex that way. But if you have any question, if you like this video, you can leave a comment. And if you have any specific request, you can still use my email to write me directly. I will answer all the questions. And thank you for your attention and see you next time. Goodbye.